First, let's look at the part of this forest. There is an interjection interspecific competition. It is a survival battle where different species compete for the same limited resource, such as sunlight, physical space, and nutrients. One of the species of tree there are Cryobalanops aromatica. They will shed its bark because it is actively competing against climbing vines and epiphytes by denying them a stable surface to grow on, thus making other trees do not get too close to them. If the tree loses these competitions and become overgrown, it may struggle to get enough light or even collapse under the weight of the other plants. This arm race ensures that only the most resilient and well-adapted plants to survive in a crowded environment.
Moreover, the interaction you see in the image is a classic example of commensalism involving epiphytes, which is plants that grow on other plants. In this relationship, the ferns growing on the palm trunk benefit significantly, while the host tree remain unaffected. Because the forest floor can be quite dark, these ferns use the height of the palm tree as a ladder to reach better sunlight for photosynthesis. It is important to note that these are not parasites. They do not steal nutrient or water from the tree's internal system. Instead, they use their roots to anchor themselves to the bark and catch falling rain and organic debris to survive. Essentially, the palms provide the real estate and the fern gain a sunny home without giving or taking anything back from the host.
Now, let's look at the next interaction. This interaction involving the moss on the boulders is a form of commensalism that plays a vital role in the forest microecosystem. The moss uses the hard, stable surface of the rocks as a substrate to anchor itself, allowing it to grow in a location where most plants with deep roots cannot survive. While the rock is unaffected, the moss benefits by gaining a home that is often damp and shaded. This layer of moss acts like a natural biological sponge, trapping rainwater and forest humidity against the rock surface. This creates a moist microenvironment that benefits the surrounding fen palms and soil, helping to regulate the temperature and moisture levels on the forest floor during dry periods.
In this second image, the interaction is quite different from the first. This is an example of saprophytism or decomposition. The mushrooms you see growing on the fallen log are saprotroph, which are organisms that feed on dead organic matter. Unlike the ferns in the previous photo that were just using the tree for height, these fungi are actively breaking down the dead wood. They secrete powerful enzymes that decompose the complex carbohydrate like cellulose and lignin in the log, turning the wood back into simple nutrients. This process is vital for the ecosystem because it recycles those nutrients back into the soil, making them available for the surrounding grass and trees to grow. While the log is being consumed, the interaction is beneficial to the environment as a whole, acting as nature's primary recycling system.
Last but not least, the relationship between Melastoma malabathricum and the Armadillo bee is a specialized example of resource for service mutualism. The process starts when the plants produce nectar to lure the pollinator to its favorite petals. As the bee learns to consume this nectar, its busy body naturally attracts and collects sticky pollen grains from the flower's unique stamens. While the bee gains their protein and nourish to its colony, the plant gains a mobile delivery system. As the bee travels to the next plant, it wraps that pollen onto the receptive stigma, facilitating cross-pollination. This targeted transport is far more efficient than relying on the vine, allowing the plant to successfully reproduce while the bee stays energized for its foraging journey.
So the first experiment that we had done was basic concepts of statistic and variation. First, let's look at the introduction. Statistics is a branch of applied mathematics that involves the collection, description, analysis, and interpretation of data drawn from a sample of a larger population. Statistical sampling is used in medicines, finance, marketing, and many other fields to increase understanding and inform decision-making. Standard deviation and variance are types of statistical properties that measures dispersion of the central tendency, most commonly the arithmetic mean. They are descriptive statistics that measures variability around a mean for continuous data. The greater the standard deviation and variance of a particular set of scores, the more spread out the observation or data points are around the mean. The objective of this experiment are, first, to calculate and describe the following descriptive statistic, which is mean, variance, and standard deviation. Second, to understand the concept of correlation. Lastly, is to determine the relationship between two variables. This is the materials that we use in this experiment, which are leaf, ruler, weighing balance, and draft paper.
Next, we have 3 steps of procedure. First one, 10 leaves were taken and their length and width were measured and recorded. The mean, variance, and standard deviation of the length and width of all 10 leaves were calculated. The data were tabulated on graph paper. Second step, another 10 leaves were taken and the same procedure was repeated. This process carried out for another 3 sets of leaves. The result were tabulated on table given. 3.1. The data from all 30 leaves were used to plot a graph of leaf length versus leaf width on a graph paper. The relationship between leaf length and leaf width was examined. It was determined whether a relationship existed and, if so, what kind of relationship it was, whether linear, straight line, curve, or another pattern. The relationship was then discussed.
Okay, I would like to explain the results for this experiment, and we have total in 3 sets, and for each set we are calculating the leaf length in centimeter, leaf width in centimeter, and leaf weight in gram. For set 1, we are calculating the leaf length in centimeter, and we got mean is 14.1, variance is 16.14, and standard deviation is 1.34. Next, we are calculating the leaf width in centimeter, and we got mean is 6.2, variance is 9.91, and standard deviation is 1.05. The last calculation in set 1 is leaf weight in gram. So we got mean is 2.22, variance is 0.2841, and standard deviation is 0.1777. For set 2, we're calculating the same thing, and we start with leaf length in centimeter. So we got mean is 8.44, variance is 16.04, and standard deviation is 1.34. Next, we are calculating the leaf width in centimeter, and we got mean is 3.52, variance is 5.32, and standard deviation is 0.77. The last calculation in set 2 is leaf weight in gram. So we got mean is 2.32, variance is 0.14, and standard deviation is 0.125. For last set, which is set 3, we are still calculating the same thing, and we start with leaf length in centimeter. So we got mean is 13.97, variance is 23.70, and standard deviation is 1.62. Next, we are calculating the leaf width in centimeter, and we got mean is 6.27, variance is 11.71, and standard deviation is 1.14. So we reach at the last calculation in set 3, and last but not least, we are calculating the leaf weight in gram. So we got mean is 9.31, variance is 0.61, and lastly, standard deviation is 0.26. Okay, before I pass to the next person, I would like to conclude for results parts, where we combine all 3 set data into one graph to show the relationship between leaf length and leaf width. From this graph, you can see it reveals a positive correlation, meaning that as the length of the leaf increases, its width tends to increase as well. This pattern suggests that leaf growth is proportional in both dimensions. However, the exact slope and strength of the correlation may vary depending on species, age, and ecological conditions. Referring to the 10 types of leaf that we use for this experiment, all show the same results of increasing proportionally. So that's all from me. Let's pass to the next person.
This experiment examined the relationship between leaf length and width using basic statistic, which is the mean, variance, and standard deviation across 3 types of leaf. The mean values show that the species with longer leaves generally had wider leaves, indicating a positive relationship between length and width. Variation in leaf size may be influenced by environmental condition, growth factor, and leaf age. Leaf length and width are ecologically important because they relate to plant adaptation and photosynthetic efficiency, with larger surface areas improving light absorption. The experiment followed careful procedures using healthy, measured leaves and consistent measurements method to ensure reliable results. Overall, the statistical analysis confirmed a clear relationship between leaf length and width and highlighted variation among different leaf types. Mean leaf length varies among the 3 sets due to differences in plant species and environmental condition. Lower leaves tend to be larger because they receive more nutrients and water, while upper leaves are more exposed to sunlight. Environmental factors such as soil, nutrients, water, temperature, and light influence leaf size. Mean leaf width is similar for sets 1 and 3, while set 2 is slightly smaller. Leaf width and growth depend on nutrient availability, especially nitrogen, phosphorus, and potassium. Based on this question, variance measures how spread out values are from the mean. Leaves from different parts of the stem shows different length and width. Each set had 10 leaves with total length and width varying among sets. Variance value confirm that the leaf size differs, helping us to understand the data clearly. It is necessary to have different sets of measurements for the measurement of leaf width and length because it can record each of the growth for each plant and decide whether it has same characteristic. It also helps to obtain record for the average of the growth in plants.
The last part is conclusion. In conclusion, the objective of this experiment were achieved. The mean, variance, and standard deviation were calculated to describe the data distribution, while correlation analysis was used to determine the relationship between 2 variables. This experiment improved understanding of basic statistical analysis and data interpretation. Let's move on to experiment 2, which is factors that affect the distribution and abundance of organisms. Next, the introduction. Understanding ecosystem dynamics requires examining the interaction between organisms and their environment. Both biotic factors, such as competition, predation, and interaction among living organisms, and abiotic factors, including temperature, rainfall, and other physical conditions, influence the distribution and abundance of organisms in an ecosystem. All living organisms present in an area make up the biotic component, while individuals of the same species living together form a population. Studying this interaction explain how ecosystems function and how environmental factors shape community structure and population patterns. In this experiment, we have four objectives, which is to introduce the simple concept of quadrant, to enhance the power of observation, to record the difference in plants distribution within the quadrant, to inter from natural observation the possible factor that affect distribution and abundance of organisms. The material that we use in this experiment is raffia strip, measurement tape, stick, scissor, and notebook.
4.0 Procedure. Different types of plants at backyard or faculty building will be the area under study. First quadrant of 1 meter times 1 meter at each forest type is made. Multiple quadrant samples is performed throughout the habitat at several random locations. All three stands that are found inside the quadrant is observed and take note. Data is used to estimate the population size and population density within the entire habitat.
So I will explain the results for this experiment. For your information, we did this experiment at the backyard of faculty building. We just have one quadrant, but my group found 7 type species of plants. So for first plant, the scientific name is Cyperus mindorensis. The total number of individuals is 111 and density is 28.46%. The second plant, the scientific name is Theodotis corymbosa. The total number of individuals is 11 and density is 2.82%. The third plant, the scientific name is Hemigraphis alternata. The total number of individuals is 111 and the density is 28.46%. For fourth plant, the scientific name is Phyllanthus urinaria. The total number of individuals is 10 and the density is 2.56%. The next plant is, the scientific name is Emilia centrifolia. The number of individuals is 4 and the density is 1.03%. For sixth plant, the scientific name is Spermacoche ochomoides. The total number of individuals is 23 and density 5.90%. The last plant, the scientific name is Chrysophogon aciculatus. The total number of individuals is 120 and density 30.77. So the total number of individuals for all plants is 390 and the total number of density for all plants is 100%. So that's all from me for the results part, and we move to the discussion. Thank you.
Now, let's look at the poster questions. For question 1, define the term quadrant in practice. A quadrant is a standardized area used in ecological research to sample and study a specific habitat. It helps assess the distribution and abundance of species within the designated area, providing a systematic and replicable method for ecological studies. For the second questions, what are the two basic requirements of all the quadrant techniques? The two basic requirements of all the quadrant techniques are standardization and randomization or systematic placement. The third questions, what will be the advantages and disadvantages of using a larger or smaller quadrant to survey the sedentary organism? For larger quadrant, the advantages are comprehensive sampling reduce the edge effects, while the disadvantages are resource intensive and less precision. For smaller quadrant, the advantages are higher precision and resource intensive, while the disadvantages are risk of missing rare species and greater edge effects. Next, for question 4. Problems with spatial scale often make it difficult to directly estimate average densities in habitats such as large, forested areas, in aquatic habitats where one cannot directly see the animals or plants of interest. Explain the issues concern ecologists in conducting the sampling method. So the sampling method is more known to be a direct approach that involves counting the number of organisms with interest at a specific designated area. However, there will be problems at each sampling trial. Firstly, the sampling method is used to collect the needed data. Secondly, incorrect selection error, as the ecologist may have a specific interest, but the wrong selection may lead to an imperfect data. Lastly, based on how to minimize the number of samples that they will take accurately. Consequence, the ecologists have developed several basic sampling approaches to deal with organisms. For the last questions, why is there a difference in the number of plant species in the quadrant? Hypnotize or make your own assumption. Discuss the factor that will have caused this type of distribution. Environmental heterogeneity, which results in varying habitats due to variation in soil, moisture, light, and microclimate, is responsible for the variance in plant species within a quadrant. Distribution is also influenced by interactions between species and successional phases, which is early succession favors pioneers, whereas competitive exclusion reduces the species richness. Factors such as sampling effort, historical disruptions, and dispersal constraints influence the existence and distributions of species in the studied region. Apprehending these variables facilitates the interpretation of plant community dynamics and inform the development of ecological sampling plans. Moving on, let's look at the discussions. For our ecological study, we utilize quadrant sampling, which is a standardized method essential for providing an unbiased look at plant populations. In this experiment, we established 1 meter times 1 meter quadrats across different places by placing this quadrat at multiple random locations throughout the habitat. We were able to record 3 stands, species composition, and density. These data serve as the foundation for estimating the total populations, size, and density across the entire ecosystem. Our results reveal a significant variation in species distribution. We found that Chrysophogon aciculatus was the most dominant species, boasting the highest density at 30.77%. On the other end of the spectrum, species like Emilia sonquifolia and Phyllanthus urinaria recorded much lower densities at 1.03% and 2.56% respectively. We also noted a high plum distribution for species like Piperus mindornensis and Hemigraphis alternata, both sitting at 28.46%. In summary, our quadrat sampling successfully identified density trends in plant distribution. The uneven distribution we observe is a direct result of environmental heterogeneity, which is the mix of abiotic microhabitats and biotic interaction that define the health and diversity of this place.
Conclusion. Through this activity, the concept of a quadrat was successfully introduced as a simple and effective tool for studying plant population in a given area. Careful observation within the quadrat enhanced observational skill and allowed for accurate recording and differences in plant distribution. The variation in the number and types of plants observed suggests that the organisms are not evenly distributed in nature. From this observation, it can be inferred that factors such as light availability, soil condition, moisture, and competition may influence the distribution and abundance of plant. Overall, the use of quadrats helps in understanding how environmental factors affect living organisms in their natural habitat.